Thanks for the opportunity. So my lab is interested in understanding the fundamental biology of heart precursor cells called cardiac progenitor cells, and which serve as the building blocks to make the heart during heart development. So today I'm going to share with you some of our recent findings that demonstrated the existence of self-renewing cardiac progenitor cells. So stem cells are multipotent, meaning that they can give rise to different cell types, and also they are capable of self-replication, which is critical to determine the number of the product cells, which are progeny cells. And this self-replication is regulated by the niche cells that serve as the cellular environment to control the number of the stem cells. Since stem cells are closely associated with a number of disease and development and regeneration processes, it's critical to understand where these niche cells and stem cells are present and how they are regulated. So as many of you know, that stem cells have been discovered in a number of the tissues and organs. However, it's unknown whether such cells are present in cardiovascular system. So I'm going to briefly go over how heart is generated. Heart is the first organ to form in mammals. So after fertilization, So after fertilization, the fertilized egg forms the structure called blastocyst. And this blastocyst contains the inner cell mass, which we call embryonic stem cells. And this inner cell mass, embryonic stem cells, can give rise to entire organism. And subset of this embryonic stem cells actually become early cardiac progenitor cells expressing the protein MASP1. And nowadays, we can easily isolate these embryonic stem cells, and then we can culture in a dish. And subset of these early cardiac progenitor cells are further become the late cardiac progenitor cells expressing the protein islet 1 here. And at this stage, these cells are pretty much committed, so they tend to become cardiac muscle cells. And if you genetically trace this, as you see here, if you trace these cardiac progenitor cells, they are all contributed to become the heart cells specifically. And this process can be recapitulated in vitro. We learned a lot of developmental biology for the past decades. So it really allows us to recapitulate this this in vivo process. So it's very important. So it's very important to understand the how these cardiac progenitor cell numbers are controlled, because you're looking at this wild type embryo, and this is the heart. And if you have low numbers of cardiac progenitor cells, often you get very small, very hypoplastic heart. However, if you have too many, then you get gigantic heart. So it has to be precisely controlled. And this process is really highly conserved. So you can even see in the Xenopus embryo, this is the heart. And if you have too many cardiac progenitor cells, you can eventually make the entire heart into the entire head into the heart muscles. So we are very interested in that if there is self-renewing cardiac progenitor cells are present in the cardiovascular system. So to address this question, we deleted the conserved sulfate regulator called NUM, specifically in early cardiac progenitor cells. And the way we do is that using the conventional Cree locksmith system. So this Cree is expressed in the early cardiac progenitor cell locus, and then that mediate the recombination of this gene called NUM, so that you get cardiac progenitor specific deletion of the NUM. And it turns out that if the deletion resulted in very small heart, which is accompanied by the very small PA2, which is the second pharyngeal arch. And it looks like, and this PA2 is the non-cardiac tissue, which is located in the back of this developing heart. And overall phenotype appears to be very much confined to these two PA2 and then small heart. So we took a closer look by making sections, and it turns out that this wild type embryos, wild type this PA2 structure contains the dense cluster of cells here, however, which are continuous to the heart. So this is the heart, and this is the PA2 structure. However, these cells were pretty much depleted in the mutant cells. So that suggests that perhaps this cells cluster could be a cell source to make the heart. But it's very interesting that this cluster of cells do not express the heart genes, which is NKX 2.5. However, they are somehow very continuous. And moreover, we found that we actually, in utero, we try to detect the proliferating expanding cells by adding this EDU, which is DNA analog, which is incorporated into dividing cells, which allow us to see these expanding cells. And we found that these EDU positive cells are pretty much concentrated in PA2, but not in the heart. So we trace these cells to test if these cells are indeed self-renewing cardiac progenitor cells. To do this, we pulse this embryo at early stages and then trace them. And after 2 hours, we found that these expanding cells in the PA2 here and there, however, none in the heart. However, after 4 hours, they just expand greatly here in the PA2 structure. And after 8 hours, they migrate out to the heart. And eventually, you see a lot of EDU positive cells in the heart, suggesting that indeed these cells are renewing cardiac progenitor cells giving rise to the heart. And to directly test this, we cut up this second pharyngeal arch cell, the structure, and then cultured it in a dish. And it turns out that after 0 days, they keep on expanding inside this structure. And they migrate some of them actually migrate out, and eventually they form the sheet of beating cardiomyocytes after 7 days. So this really tells that this second pharyngeal arch actually may serve as niche cells to promote their proliferation and then block their differentiation into cardiomyocytes. However, if you remove these PA2 cells, that they slowly become these cardiomyocytes after 7 days, and then eventually they make the beating heart muscle. So this really suggests that these PA2 cells are niche cells that control the self-renewal of cardiac progenitor cells. So go back to our earlier phenotype that we deleted this NUM, and we saw the hypoplastic heart and PA2 structure. But we investigate this further. But the problem is that if you generate this embryo, they are lethal at very early stages. So we developed the new way to rescue this, which is we established the embryonic stem cells from this cross, and then these are mutant ES cells, and then we deliver directly into normal blastocyst. In that case, you get the perfectly normal chimeric embryos. However, you can still see these red cells are mutant cells. You can still trace these mutant cells. And it turns out that if you look at the normal PA2 sections, there are a lot of proliferating cells indicated in white. That was perfectly rescued in the chimeras. However, if you take a closer look here, that in wild-type cells, unlike this normal cells, these chimera cells, that you're seeing this red cells are mutant cells. They never express this proliferation marker. So we tested it with a number of embryos, a number of chimeric embryos, and then we found that these cells never become this self-renewing fate. So this is our current model, that this is developing embryo. This is the second pharyngeal arch, and these cells kind of keep on self-renewing and then adding to these progenitor cells, and then migrate to the heart, and then become these heart muscle cells, and that could be a potential mechanism to make the heart in fetal development. So just to summarize that, we found that this second pharyngeal arch contains, PA2 contains the self-renewing heart precursor cells, and that PA2 actually serve as the microcellular environment, which is niche cells, serve as niche cells to maintain them in undifferentiated state. And then this self-renewal is regulated by the protein NAM. And we are in the process of figuring out what factors are secreted from PA2 structure, and then how they interact. And that's our next goal. So actually, I'm not going to get into this, the potential of these cardiac progenitor cells, but I can tell you that these cardiac progenitor cells have just tremendous regenerative potential, and we hope that we can actually use these cells to maintain in a dish and then just use for this cell-based therapy or disease modeling. And this is work in progress, and hopefully I can share it with you sometime later, next time, So I'll stop here, and these are my members. They work really hard. So I'll stop here. Yeah.